Hello, everyone. I would like to start by thanking the organizers for the opportunity to be here today, and also to thank you for being present in today's session. My talk is about my PhD topic, which focuses on studying the immune system dysfunction in space. I will start by sharing a bit of my background with you, and I will then follow with explaining the design of a project. I also wanted to share with you my personal perspective on what pursuing a PhD in space studies is like, and what challenges there might be, but also small victories and future work directions. So my background is in radiotherapy. I'm a radiation therapist, but in 2017, because I wanted to focus on research, I followed a master's degree in molecular medicine and oncology, and I wanted to focus my research on radiobiology, particularly on the role of mitochondria in the non-targeted effects of radiation. And in 2020, I started my PhD at SCK CEN in Belgium, together with the University of Ghent. And the topic I chose was in space. So I was really interested in space radiobiology and pursued this PhD in space immunology. In space, there is an exposure to stressors, such as altered gravity, radiation, or increased levels of stress. And these exposures causes the body to adapt, and some of these adaptations turn into dysfunction of some systems, like, for example, the immune system. I will talk about some of these alterations in the immune system in a bit, but first, I want to explain why we are focusing our work on T cells. So T cells are important regulators of the immune response. They not only kill infected cells, but they also activate other immune cells, and they are key for the memory immunity. Indeed, in space, the space immune dysfunction has been described through various studies in the form of, for example, reduced T cell activity, an alteration of the cellular compartment levels. There is also some evidence of increased reactivity. For example, viruses are reactivated while in space missions, or there is an increased allergy levels, and there seems to be an interaction already described between microgravity and radiation. But what is still not clear is what is causing it, namely, which of the stressors. Is it one of them, or are they working together in producing this dysfunction? And what we would like to do is to pinpoint this mechanism, and maybe this will help us think about some countermeasures. How we will tackle this? First, we will use UCAT cells. In the first phase of our project, we will stimulate them and expose them to simulated space conditions. So we will stimulate them first, and then we will expose them to simulated space conditions, such as altered gravity, ionizing radiation, and also stress hormones. And we will do this in combination or alone. And we will look at IL-2 levels exhibited by these cells, either in the supernatants of these cells through colorimetric assays, for example, but we will also want to validate these findings looking at the gene level and to the IL-2 gene expression. And this will help us for our next stage of our project, which will be with the CD4 positive T cells that we will collect from healthy donors. And this will also be stimulated and in the same manner exposed to simulated space conditions. And then with these cells, we want to go a little further. So we want to analyze the profiles, the differentiation profiles, of course, also the cytokine levels that they release, but we will go a layer deeper in complexity, and we will do some proteomics and transcriptomic studies in the pursuit of this mechanistic approach. So how do we tackle this? How do we study space immune dysfunction? And this is the concept. I showed you the concept, but now how do we achieve it? So first, the URCAT cells, why are we using them? So URCAT cells are derived from the leukemia patient leukocytes, and they are fairly lots used in the studies on T-cell activation, and they express high levels of IL-2. And why IL-2? Well, IL-2 plays a role in the infection response. It's very important for T-cell differentiation into either effector cells or memory cells. And URCAT cells are known to produce them in large amounts. And we believe that this will help us guide our next stage with the CD4 positive T cells. So we want to establish a trend of the exposure to the stressors that will then guide us in the CD4 positive T cells. What about alter gravity? To expose the cells to alter gravity, we will make use of the random positioning machines that we have here at SCK CEN. So a random positioning machine works by continuously changing the orientation of an object, in this case our sample, to relative to the gravity vector. And these changes are faster than the response of the model that we are studying to the gravity. So these are ideal to use in biological system studies. And they also allow us to expose our samples to altered gravity. So we can simulate Moon and Mars gravity. We are interested in studying partial gravity in these cells and exposure response of these cells. And we are looking into Moon and Mars levels in particular. Next is stress. So why will we stress cells? In space, the space missions are highly in high level pressure missions. So we believe that conveying these missions are in a high level of stress constant. This will also add up to the fact that they are in confinement or isolated from Earth. And we believe that there is a high level of cortisol present in the system due to this. So we can simulate this on Earth by putting volunteers in analog missions, in isolation analog missions, but we can also do it in vitro. And how do we do it? We expose our cells through hydrocortisone dissolved in the media. And we believe that this induces a relevant stress response that can be analog to what astronauts experience in space. Last, but certainly not the least, radiation. So space radiation. As you know, radiation in space is composed of galactic cosmic radiation that comes from outside our solar system. And it's mainly composed of protons and also other highly charged particles, like carbon or iron. And it also is composed of the solar particle events that come from the sun in our solar system. They are accelerated protons from coronal mass ejections, for example, and also some other highly charged particles. How do we simulate radiation, space radiation here on Earth? We need to find a way to accelerate these particles and expose our samples to them. We can do this using cyclotrons. These are then accelerating the particles, for example, carbon ions or iron ions, and also protons. And here in the images, we can see some of these irradiation facilities. So on the picture on the right is in France, the Ganil accelerator, accelerator where we have been to expose our samples to carbon ions. And the picture on the left shows some samples ready to be irradiated with protons in the KVI in the Netherlands. Now, the road to get a PhD. This is actually a road in Belgium, and I find that it is a good depiction of the journey of a PhD is. this is where I am. So I just finished my first year, and it was a full and eventful year indeed. The first challenge I want to share with you is immunology. So the team in itself, immunology is very challenging because the cells are very sensitive and the cytokine levels are often transient. So any variation on the conditions can induce different results. For example, I can show you, this is a setup for an ELISA assay that I performed for IL-2. And in one day, I couldn't find any IL-2 levels, and exactly the same samples on the next day, exactly the same ELISA protocol. I can already see by the change in color that indeed there is IL-2 present in the media. So this means that a lot of the year was spent on optimizing these assays in order to obtain the ideal setup and conditions to perform them. Another thing we need to tackle is the space-relevant radiation, like I mentioned. So we need, in order to expose this, we need to go to the facilities. There are a series of facilities in Europe. We can apply for beam time, but going on a radiation campaign is a challenge. It requires a lot of planning, and we are basically traveling with a mobile lab to these locations. So planning every detail, including a series of contingency plans, is key. However, these are unique opportunities because not only we can expose our samples to simulate space radiation, but also we can prepare in one go several assays, several samples, which of course translates into data. And this is what we want. We want data. I have been present in 4 radiation campaigns this past year, and thanks to them, I can now show you some preliminary findings. So indeed, after exposing the samples to carbon ions in the France accelerator in Ganil, I can show you some interesting findings we obtained. So we are looking at the cell activity in the form of IL-2 release, as I explained, in UCAT cells. And what we can see is that when we expose them to stress, so to hydrocortisone, high or low concentrations in combination with the carbon radiation, we see a decrease of IL-2 release. But if we expose the cells to a combination of the moon gravity levels and radiation, we see an increase in this activity. So indeed, these are interesting findings, but they raise a lot of questions. What does this mean? So of course, we need to pursue this further. We want to continue this analysis. We are looking into validating these findings with our qPCR that we are now optimizing, but we also want to perform more irradiation campaigns to have more data points. Also, another types of radiation, we are now looking into irradiating our samples with iron, but also other gravity levels, and also some more combinations, because we were only able to do combinations of 2, but we want to do the combinations of the 3 stressors at the same time. And of course, we want to look into this with CD4 positive T cells. That's the next step on the road. Now, I like to finish by sharing some take-home messages with you. Indeed, space research has a lot of potential, not only because we are studying to improve astronauts' health, but we also know that everything we can find can and will be used for Earth applications. I hope I showed you that it's also possible to simulate space conditions on Earth and how we can tackle this challenge to perform our studies. And I think I showed you that a PhD is not straightforward. It will have some challenges, but I believe that all this work is what the PhD is about. It's not about the end point, so the project design and the idea. is nice, but the PhD is actually the road there and not the destination. Thank you for listening, and I'm here to answer some questions if you have them.